Chromatography is used to separate components based on their volatilities, which based on their boiling point. Gas is called carrier gas, and typical carrier gas is helium or nitrogen. There are two types of chromatography, which are the first one is gas liquid chromatography and the second one is gas solid chromatography. In gas chromatography, components of vaporized sample are separated as the result of partitions between a mobile gaseous phase and liquid or solid stationary phase that held in the column. Sample was injected at the injector and vaporized into the chromatographic column, transported through the column by the flow of inert gases of mobile phase. As the sample passes through the column, it has been separated and detected by the instrument or detector.
Assalamualaikum and hi, my name is Sarah Safi binti Hassan and I will present the methodology, the reagents, samples and standards. Dichloromethane, methanol, absolute ethanol, pure water, lychloride EN resins, prepacked in 200 mg cartridges, 3 ml total of volume. The chemical standards, the internal standard solution that contain 4-hydroxy, 4-methyl-2-pentanol and 2-octanol, both at 300 per gram of dichloromethane. The BHA, which is 3,3-butyl 4-hydroxy anisole, contain 10 mg of this compound per gram of ethanol. A 5-year-old red wine, a young white wine, and a rose wine. Gas chromatography, mass spectrometry. A Star 3400 CX gas chromatograph fitted to a Saturn IV electronic impact ion trap mass spectrometer from Varian was used. The column was DB-WAX-ETR 60 meter times 0.5 mm with 0.5 mm film thickness and was preceded by a 3 × 30.32 mm uncoated precolumn. The carrier was helium at 1 ml per minute. The chromatographic oven is initially 40 degrees Celsius for 5 minutes and then was raised to 230 degrees Celsius at 2 degrees Celsius per minute. A 1093 SPI, which is septum equipped programmable injector from Varian, was used. The initial temperature of this injector was 30 degrees Celsius for 0.6 minutes and was then raised to 230 degrees Celsius at 200 degrees Celsius per minute. Then 3 ml of sample were injected. At 35-220 m per sec, mass range was recorded, and the extracted ion chromatograms were taken for quantitation by peak area. Ion peaks were taken for quantitation. Next, determination of the distribution coefficient of solid-liquid systems. An exact weight of the sorbent, which is 0.12 gram, was placed inside a glass vial together with a 50 ml volume of wine fortified with 2 mg per liter of some selected compounds. The vials were shaken softly for 24 hours. After this, 10 ml of the extracted wine was transferred to a 15 ml centrifuge tube containing 3.3 gram of NH42 SO4 plus 20 ml of the internal standard solution and 0.5 ml of dichloromethane. The tubes were closed. It has been shaken gently for 45 minutes, centrifuged, and the organic phases analyzed by GCMS. The relative areas were compared to those obtained in the direct analysis of dichloromethane extracts, which is 10 ml wine plus 3.3 gram of NH42SO4 plus 20 ml of the internal standard solution and 0.5 of dichloromethane from the fortified wines. Phase ratio and hold-up volumes were directly measured by weighing the chromatographic beds before and after the addition of the necessary amount of mobile phase to form the bed, and after the expulsion of interstitial liquid by a flow of air. The number of plates, N, was estimated from the breakthrough curves built in frontal elution experiments. There are four proposed methods. The first one is cartridge conditioning. The cartridge with the sorbent were placed in the extraction system and rinsed with 4 ml of dichloromethane, 4 ml of methanol, and finally with 4 ml of a water-ethanol mixture, 12%. The next one is sample loading. The 50 ml of wine containing 25 ml of BHA solution were passed through the SPE cartridge at around 2 ml per minute. After this, the sorbent was dried by letting air pass through it. The third one is elution. Analytes were recovered by elution within 1.3 ml of dichloromethane. 25 microliters of the SI solution were added over the eluted sample. The mixture was then hermetically kept and stored at 225 degrees Celsius until the GCMS analysis. And the fourth one is calibration. Calibration graphs were prepared by the GCMS analysis of dichloromethane solutions containing known amounts of the standards and of the internal standards. Lastly, the method validation. The recovery of the SPE process was determined by the analysis of the 3 wine samples before and after the SPE. The reproducibility of the method was determined by replicate analysis of wines 1 and 2 on 3 different days. The existence of matrix effect was checked by the replicate analysis of wines 1, 2, and 3 and of those wines piped with known amounts of analytes.
Assalamualaikum and hi. My name is Aina Baiduri binti Muhammad Nizam, and I am going to talk about the analysis of this experiment. This experiment is to trace the volatile compounds in wine using solid phase extraction and gas chromatography with mass spectrometric detection. So based on the results, we can see that vanillin has low peaks in the graph. This is because vanillin has high volatility. Compound that has high volatility is the compound that has high boiling points. Vanillin's boiling point is 285 degrees Celsius, which is very high. This caused vanillin to travel much more slower during the gas chromatography. Vanillin is also a polar molecule because it has CO and also OH bonds in it. Polar molecules tend to move much more slower during the separation, thus makes vanillin, which is a polar molecule, move much more slower than other compounds during the separation of wine in the gas chromatography. That's all from me. Thank you.
As for the conclusion, the proposed method makes it possible to determine the wide range of the wine aromas, such as vanillin as a volatile phenols, present at minor and trans concentration after a single SPE isolation process, while the vanillin has a low case for the low reproducibility, as for its poor and erratic recoveries. The analytical properties of the method are satisfactory for the purpose of the study for the wine aroma.
Assalamualaikum and hi. My name is Aina Baiduri binti Muhammad Nizam, and in this video, me and my group mates will be talking about the determination of vanillin using HPLC in alcohol. So, high performance liquid chromatography, or known as HPLC, is a chromatographic method that is used to separate a mixture of compound by utilizing a liquid mobile phase. HPLC is very versatile because of its ability to easily separate a wide variety of chemical mixture, which includes non-volatile and thermally unstable compounds. In this experiment, HPLC is used for vanillin determination in alcohol. I'm going to pass this video to the next person to continue this video. Thank you.
In performing high-performance liquid chromatography, the first step is by running the software as a preparation before put the sample into the instrument. The second one is for the sample preparation. Firstly, sample has been transferred into vial by using shrink and bin filter. Secondly, mobile phase has been filtered by using membrane filter. The next one is inject the sample and running the instrument. As the result has been recorded, it is also been printed.
Assalamualaikum, my name is Nur Amal Fida binti Ibrahim, and I will proceed with the analysis part. First of all, the elution time of the vanillin is taken between 7 minutes to 36 minutes. Then, the average flow rate of vanillin is 1 ml per minute. The authors have mixed the solvent with the methanol, and also the majority of the authors have agreed the optimum wavelength of the vanillin is 275 nanometer. The mobile phase consists of 40% of pure water and 60% of HPLC grade methanol, which is suitable for the instrument column, which is the nucleosyl C18 150 multiplied by 4.6 mm with metaguide column. The mobile gradient scheme is used and each running for 15 minutes. Then the injection volume is 20 microliter. The retention time taking only 2.21 minutes, and for the wavelength is 231 nanometer, indicating there is no interference of other compound present. The lowest detection limit is below 20 microliter as the initial injected sample. The vanillin is dissolved in mobile phase to evaluate the quality of the analytical techniques at 0.05, 0.1 and 0.15 gram per liter. Then the standard solution is added to the ethanol vanillin extract. The mean recovery for vanillin standard oscillated is between 95.8% until 100.6% and 3.2 of standard deviation, indicating the perfect result. Due to the FDA procedure, the vanilla extract can only be consist of 100 ml or milligram. The vanillin content in 6 Mexican commercial extract and 1 USA is varied, and only from the 3 of them is met the requirement of the vanilla extract, which consists of 100 milliliter or milligram.
Assalamualaikum and hi, my name is Sarah Safia and I will be present the conclusion for the simple and rapid HPLC technique for vanillin determination in alcohol extract. In conclusion, this study contributes to the establishment of easy HPLC conditions for rapid and sensitive vanillin, quantification in vanilla extract, and for routine purpose analysis. This technique can be used by vanilla extract manufacturers and consumers. By analyzing the vanillin content in some commercial products, only 3 extracts of 7 met the requirements for the vanillin content of 100 mg per 100 ml.